ladies and gentlemen. The year is slowly coming to an end, and so is our open mic series of 2025. I'm really happy that many of you came here today to listen to our distinguished panel speak about innovations in vitro models for preclinical testing. My name is Rael. I am hosting this event series with the Switzerland Innovation Park Basel area. We are operating a small office and co-working space here on Novartis campus for health tech ventures. We also have sites in Allschwil and in the canton of Jura, where we have many life sciences companies in-house. The event series, Open Mic series, has been going on for a few years and is curated by my colleague Ursula from Day One Healthcare Innovation. Ursula is director and the head behind the series. Before I hand over to you, we are, again, as usual, using Mentimeter. You will see the QR code now on the screen, so you can send in your questions via Mentimeter, and the moderator will see that during the session. Over to you, Ursula.
Thank you, Rahel, and a special thank you to everyone on site, but also online. Thanks for being here today. Last open mic of the year, but we promise we continue next year, so don't miss out. So we have a really interesting topic today. We know preclinical testing is in rapid transformation, and I'm really excited to have CSEM this time with Gilles Vedder that helped me co-curate the topic and find the best speakers. So I'm really looking forward for an exciting conversation. I know you're not here for me, you're here for them. So I'm gonna give them the stage. And please, Gilles, John, Annie, Dominic, welcome to the open mic. And we are all really looking forward to listening to you and to all your insights. Thank you.
So welcome to this exciting session from LabMice to LabTech. My name is Gilles Vedder. I am heading the research and business development at CSEM. My role is to define the roadmap and the partnership for the life sciences. CSEM is a public-private research innovation center with the main mission to transfer technology to the industry. We focus really on the technology transfer and product development to accelerate the transition between the emerging technology and the industry. We innovate at the interface between the biology and the technologies in different domains: drug testing, regenerative medicine, and diagnostic. At CSEM, we are about 630 specialists, and my life science team is located on 4 different sites, including Basel at the Swiss Innovation Park in Allschwil, but also in Neuchâtel, in Alpnach, and Landquart. We collaborate with startups, with SMEs, and biopharma industry. So I would like now to open this session with the bioconvergence. And we are in the century of the bioconvergence. The bioconvergence is a mega trend. And for a long time, the science has been divided in different disciplines. And today we have this transversal, this multidisciplinary approach that integrates biology, engineering, and artificial intelligence to address the current challenges. So we observe a revolution with the bioconvergence in healthcare and in personalized medicine. And drug testing can be personalized by human MPS, by human microphysiological system, who can replace or be in complement to animal testing. So the MPS, the microphysiological system, are advanced in vitro platform combining biology and engineering. And the biology is composed of human cells. It can be single cell, spheroid, microtissue, organoid, assembloid, depending on the application. And the engineering refer to the design and fabrication of the technical component, such as the chip or the plate. And for example, an organ on chip is an MPS, is a microphysiological system. So this MPS represents a transformation approach to study the human tissue under conditions that will mimic the physiological or the natural environment. And in the audience, we are all interested in this transformation from animal to human in preclinical drug testing. But there are, of course, many questions. Why this shift is so important? What are the main limitations on relying on animals? What are the potential of microphysiological system? And now it's my great pleasure to introduce our panelists from Roche, from Insvero and Novartis. They are distinguished leaders in the biopharma industry, representing both the tech provider and the end user. And we are extremely happy to have Annie, Jan, and Dominique with us. And I propose to start with an introduction with Annie.
Oh, I start. Okay, ladies first. Thank you. Well, first of all, thanks for the invitation, and it's really a pleasure to be here and to have a full house. It's a very engaging topic. It's a topic that's very close to my heart as well. I've been in the field for some years. It's challenging, but we do see the need, we do see the opportunities us also. Quick introduction myself. I'm the discovery area leader for nephrology at Roche in the period organization. So my group is a discovery group. We focus on kidney diseases, and we are in the cardiometabolic and immunology department. I'm a stem cell biologist by training, so I started at Roche 15 years ago. It was already quite a different landscape on how we approached biology and drug discovery at the time. My role was implementation of human stem cell derived models of diseases mainly. And I think myself, with colleagues, we quickly realized that our 2D simple static models were simply not good enough. And we needed to augment the biology, and we started to invest in collaboration with academic groups, with CROs, and then through this career, I could work in discovery, in mechanistic safety, so really learning about the efficacy and the toxicity of drugs and how we can use and leverage these bioengineered human models, or MPSCs. I worked also for philanthropic organization for some years, back at Roche now to continue developing these systems with the community. Thank you.
Annie. Jan?
Yeah, thanks for having me. My name is Jan Lichtenberg. I'm the co-founder and CEO of Insvero. We are a Zurich-based company focused on human in vitro models. We started 15 years ago to develop sand grain size 3D approximation of different tissues that we have in the human body for safety and efficacy testing. Our focus has always been on scalability and the sort of democratization of this technology, because we believe if it's easy to use, if it's easy to implement, if it's easy to scale up, more and more researchers are going to use this. We're now about 100 people. We have produced about 1 million of these sand grain sized models last year. So it's a technology that found its way into the application, into the drug discovery industry, but also into cosmetics, into chemical industry, and also into academic research. And we're not the only company in Switzerland in this space. I think Switzerland is a very strong marketplace for these technologies, starting with organizations like CSEM that develop some of the sort of groundbreaking technologies, but then also a lot of startups that have commercialized different flavors of human in vitro models. And it's great to have the discussion here today.
Thanks. Dominique?
First, also, thanks for having me and a nice intro. My name is Dominique Köpfner. I work here for Novartis on the campus. By training, I'm a molecular biologist and geneticist. And I got my job here 20 years ago with the focus of finding novel entry points to tackle diseases. And we're using genetic methodologies. And roughly, we have used a variety of different methods for in vitro screens. And roughly since the inaugural papers by Jinek and Doudna and Charpentier 10 years ago, it's pretty much all focused on using the CRISPR methodology to manipulate the genome to learn more about diseases. Now, one thing we've learned in the last 10 years leveraging the CRISPR methodology is that the method to really manipulate the genome to knock out genes or to overexpress genes is really usually working very well. There's sometimes a couple of hurdles, but eventually it works well. But what we learned the hard way is it's very, very important in which biological context you ask your biological question. It really doesn't make a lot of sense to ask things related to neurodegeneration when you're actually manipulating cancer cells. And so learning the hard way that in order to get good biological answer, you need to have the right biological system. We have really significantly been investing and upping our game, coming from cell lines in 2D on plastic to 3D cultures, to iPS-derived systems, to primary cells, to organoids. And it's getting things a lot more complex, but eventually also much more rewarding and insightful, and it translates much better towards the clinic.
Thank you. As you can see, we are in good hands. We have really expert. And just a bit to set the stage, I propose to start with one question with each of our panelists, a bit more general question. And I will start with you, Jan. How would you explain to a general audience why this shift from animal models to MPS is so important, and how the technology like gene therapy, cell therapy accelerate or reinforce this importance?
So animal models have been a workhorse for the industry for many decades, and a lot of innovations in the field are based on these animal models, but they have strong limitations, and that is they are not human, right? And we are developing drugs for human. And as we are working on more and more precision treatments that are relying on cell and gene therapy, RNA therapy, these therapies rely on human biology to work. So if we don't have this available and we are trying to approximate it using an animal model, then we are typically missing out. The metabolism is slightly different, the immune response is slightly different, maybe a specific target sequence is not really expressed in that model, and that leads us down the wrong way. To give you an example, when paracetamol, acetaminophen, had been tested for safety in probably 50, 60 years ago, it was tested on rats, and rats don't have a liver enzyme that converts that drug into a toxic byproduct, which is liver toxic for humans. We have the enzyme, the rat doesn't, and so we get wrong information, right? And this has led to this specific drug to move from an over-the-counter unregulated drug to one that is black boxed and is sold in smaller quantities down the line. And it is still one of the main reasons for liver transplantations in the US because of overdose of this specific drug. So could we have found out early on? Definitely. And this is a simple case because it's a small molecule. If we think about cell and gene therapy, we are much more focused, and if we don't have the right model in our hands, we are going to make wrong decisions. We will have less visibility on our clinical trials, and ultimately this costs time for the companies that work on this, and it costs time for the patients that are waiting for treatment. So to start with a human model from day one, in my opinion, is a key for more efficient and more successful science.
Thank you. So as mentioned, these animal models have been the foundation in preclinical testing for many years. But can you maybe, Dominique, share with us what is the main limitation on relying on mice and any type of other animals?
Well, we just heard some examples now from Jan. I mean, again, mice are not humans. They have a completely different lifestyle. Mice live for 3 years. We live for hopefully 80 years plus. Just thinking about neurodegeneration diseases that manifest themselves only after 60 years into the life cycle of a human being, to model something like this in a free-roaming animal that has a short lifespan, you see the challenge. But more importantly is also that today drug discovery very often actually doesn't start in the lab, but it starts in the hospitals and with the patients out there. Thanks to all the omics technologies and biomarkers that we have nowadays, very often it's the real world evidence that it gets collected from the patients and in the clinics that actually direct us towards some root causes of disease. And you may find mutations in the DNA, particular SNPs. Now, if you want to understand and identify if this is the cause of disease, you're completely screwed if you have to do this in animals because the genomic sequence will be very different. So you need human cells. And then, however, you have to challenge that if it's a neurodegenerative disease, you have to build a relevant system in a dish that can recapitulate this, but in a human context. And so animals definitely have their limitations, and then you bring it back into human context, but then you really have to challenge to build your system to make it relevant. And this is a process, and it's not perfect, but I think, the time is better spent to make your human system better than actually dealing with an animal model that is flawed from start and then try to back translate why did it not work.
Mm-hmm. So we see now this transition. So in terms of potential high level, what is the potential of this microphysiological system, maybe for Annie?
Yeah, I can probably add on what has been said, because it goes in the same direction. There's clearly a convergence of technologies right now that make possible a paradigm shift, in my opinion, in drug discovery. The way we think new targets, new therapeutics, what are the new avenues we can bring to patients. We used to look into mouse model of disease, to read the literature and compile evidence, and then deviate some hypotheses and test in animal models. Now, as Dominic mentioned, we have access to patient data. We are generating in real time those data with genetics, yes, genomics, yes, transcriptomics, proteomics from plasma, from tissue. So now we, instead of looking at what the mouse is telling us about a disease, a pathology, or a biology, we look into human data. That means, in terms of translatability, how we take this great idea into the clinic, we no longer need to go through an animal, maybe. Sometimes we need for some PK questions and so on. Animals are still valuable for specific aspect of drug discovery and development. But I really see that as a paradigm shift. We go from, we want to treat humans, so we start with human, and we have the different technologies to get there. And then to build this system, it can be from stem cell derivation, from explant, ex vivo tissues, and organoids. There are really a wide variety of bioengineered systems.
Yes, so thank you for this first overview. It was also mentioned by Ian that everything should begin with the needs. However, globally in the MPS, we have some limitation that can be linked to high cost, the cell source, the reproducibility, the incomplete systemic complexity, some time scale, we also mention it, and also the challenge across multiple organs. And among all these challenges, we need also to select some topic for the day. And today I propose that we go a little bit deeper in the immunology, because this is a hot question at the moment. Of course, we must go through the throughput. I think it's interesting to link the throughput with the artificial intelligence, and also a few words related to the regulation. So if we start with the immunology, the animal models are often defended because they capture the whole system immune response. But the human immune system is highly variable between individuals. And now the question is if MPS are able to reproduce this complexity and diversity, and do we have a risk of overstating these predictive powers? So I'm sure that you have all knowledge in immunology, but I know that Annie, you have a specific background in immunology. Maybe we'll start with you.
And stop me if I go too deep, because this is a really fantastic topic. And I'm not an immunologist. I have also to disclose that, because it's a very complex biology, and that's why also recreating the immune system is complex. And the need for it is obvious. I think in terms of disease, in terms of therapies we are developing, immune responses are really central to chronic diseases of any kinds, to aging. And then with immune therapies, with antibodies, we engineer molecules that are now recognized as foreign by the body, and we can develop antibodies against the drug we inject. So overall, from a biopharma perspective, being able to recreate a good immune system that is representative of human immune responses can really be transformative. You mentioned, is there a risk of overstating the predictive value. That's what you said.
Yeah.
Yes, there's always a risk of that. I think in terms of immune responses, we can both over-predict and under-predict. It really depends what we're trying to do. It really depends of what system we use. I think it's on us all to be aware of the limitations and to be transparent about limitations to avoid this overstating. So where to start? So clearly there are profound fundamental differences between species in terms of immune system and responses. So we know that mice are not humans. It's true for many diseases. I would argue it's even truer for the immune system. And even non-human primates that are much closer to humans, we also learned the hard way that they can miss out on certain toxicities in the clinic of new drugs, for example, and there are published examples of that. So there's a case for building human-relevant system of the immune systems, and they have to be human for that reason, and they have to be complex and bioengineered, because we have a tendency, I think, to think that immunity happens in blood. We say, blood cells or white blood cells. That's how we kind of understand immunology, but actually immunity happens in tissue. So if we think, first of all, first challenge, we need to recreate the complexity of all those immune tissues that are present in the body that cross-talk, cross-react together, a thymus, the lymphoid organs, and all organs in the body have their own immune cells in addition to the blood. So that's a first bioengineering challenge, in my opinion, to recreate these tissues and make them immune competent. With that first challenge comes the second challenge of immune compatibility. Think of transplantation. You need to find the best match donor recipient, otherwise you have immune rejection. When we start mixing in vitro, obviously there are some also immune reactions happening that are not desired, so we need to control for that. The gold standard right now is to have a perfect match. The same donor, we have blood and we have tissue, we combine that in vitro. Now you can imagine the logistic complications in doing that for every models assay we're trying to do. So one solution is to learn what is the minimum immune incompatibility I can afford in vitro to still be able to measure a response that is relevant and that gives me a window, an assay window for those of you familiar with the term. And solution for that is really to have collective efforts and really using perhaps also artificial intelligence to predict what this upload compatibility can be to improve the in vitro system. And then the third, probably most important, often underappreciated, is the immune diversity. Every T cell is different from the neighbor T cell in terms of naive T cells and B cells. They have their specific receptor. That is something totally unique. Like if you take liver cells, like two hepatocyte cells, they are more or less the same. But in terms of lymphocytes, they're all different and they respond differently. And this is meant to be this way to be able to respond to this infinite possibility of antigens out there. So to make a long story short, we need now to recreate and include these diversity in vitro system. And if we take a blood sample, that is our source of immune cells that we put in our system, we are for sure undersampling the diversity by multiple orders of magnitude. So will this be even possible eventually to recreate this immune diversity in a dish? I think it's an open question. I think what we've seen as potential solution is the use or recreating functional immune organs, functional thymus, functional lymph nodes. And it may sound like it's science fiction, but we see evidence that this is possible. And the goal ultimately would be to use chips, microfluidic chips, connect these organs all together and have circulating blood. So maybe not next year, but in 10 years from now, we can probably see advances. And here I would say artificial intelligence can really catalyze what we do and probably bring some focus we could not think of before. Like what if artificial intelligence tells us that out of these quadrillion of possibilities of antigen response, there are just some thousands that are actually relevant for human immune responses. And I think this is where the frontiers of bioengineered immune systems are going into.
Reaction from the two colleagues?
I think I have a more philosophical view on it, in a sense that I would divide it into an element of understanding and then a translational element. So, yes, first to understand, you really have to go deep and you have to dissect. And I think, kind of if you're if you're a native person from somewhere lived in the rainforest and the first time you see a Ferrari, you can look at the thing as long as you want. You will never understand how it works unless you start to disassemble it. And it's immensely complex. And, in that sense, it's not a mystery that the vast majority of all animal experimentation is for basic research. It's we need to understand. However, eventually, if it comes to translational aspect, and you start to understand how this immensely complex machine works, and you're just interested in one particular part. Let's say now you like the sound of the horn of the Ferrari. You don't need to build the whole car. It's just fine to use only those parts, given you understand the context in which these parts need to be in. So I would say that for translational science, it's really important to be as complex as needed, but as simple as possible. But first always comes the understanding, and then you can take a reductionist approach. But I think it's very important to really have a clear goal. What type of system do I want to build? Which type of questions do I want to ask? And sometimes it doesn't need to be very complex if it's a very simple question. And so I don't think we should recreate a real organ or a real immune system in a dish. I mean, yes, it would be fantastic, but I think pragmatically, it's perfectly fine to say, I'm interested in this particular aspect. How can I really dissect this at scale robustly?
I very much agree here.
Yes.
I think this is sort of probably the engineering approach to this, right? So so I'm an engineer by training, and when we talk to biologists, then when we hear complexity, it's typically meant to be something very good, right? The more complex, the better. As an engineer, you think exactly the opposite, right? The more complex, the more expensive, the more easy it is to fail, the more time it takes to make it and to make it work. So finding that right context of use, as Dominic said, and understanding what is the question, how is the answer supposed to look, that allows us to tailor the system in a way that it's the most efficient solution for this specific use. If you are too cheap and too simplistic on the model, then you're going to save money and time at that point, but later on you pay for it because you're going to make mistakes and you will have attrition in the clinic. If you are too complex at the early stages, you overpay and you lose time at that point, and you will not recover that later on because you've generated information that most likely nobody needs. And so having a good understanding to see how complex things need to be, and also being aware that it's typically not a single test that decides on the fate of a drug, right? It's a tiered approach where you are adding complexity to the assay while you're reducing the number of molecules that you look at. That can help us as a guiding principle. So if we talk about toxicology, an early flagging would probably not include immune competence, but before you go into the clinic, it's definitely something worth looking at. But the big difference is in that early stage, you will look at thousands of compounds, and then at the late stage, you might just look at 5 compounds. And so the extra complexity of the assay at that point is not only feasible to do, but it also makes economic sense. And then you have a system that works very well.
Can I add to this? Because I can only agree with the you don't go complex if you don't need to. I think we all agree to this. We need to be pragmatic in these approaches. The one example I believe does require this level of diversity I was referring to is when it's about predicting undesired immunogenicity of our therapies, because there we know it's a new antigen. The body has never seen that. So you need a naive response. It's not a recall response. It's not like we've never been exposed. So and we know we fail really poorly with animal models, with NHPs or non-human primates, and we learn only in the clinic. And that has a dramatic impact on multiple good drugs that we aim to develop. So that would be the one of the use cases, I think, which is very clearly you need this complexity. Otherwise, fully with you, if it's a cytokine response, more like innate immunity, smaller, a blood test can also work well, and adding some immune cells to a system in vitro will definitely work. I'm with you.
So this complexity has a direct impact on the scale-up, on the parallelization of the system, and especially for organ-on-chip system, the throughput is always coming back on the table. So we hear some critics towards organ-on-chip. So what is the current progress now to run many experiments in parallel, and how close are we to have some fully automated process with robotics to bring them at the same scale of traditional assay? Maybe let's start with you, Dominique.
So I think the field has come a long way. I mean, yes, robotics and standardized kind of products that are compatible across a vast kind of inventory of different machines that you can plug in. It's a fantastic help. Yeah. The challenge about lack of scalability, I'm not so sure I buy into this, because animal experimentation never has been high throughput, at least not in modern times. And so I actually would say that yes, obviously we would like to have things in vitro much more scalable, but in essence, if we think about it, it's a one-to-one kind of replacement of an animal experimentation. So, usually you don't need thousands. And much more, I think an in vitro system has the option to be scalable to that number. An in vivo experiment would never have. And so, yes, I mean, there's a lot of engineering nowadays in automation. And then also on the data side, you generate a lot of data, but thanks to AI, again, it's much easier to get a grip of what you generate. Yeah. It's not perfect. I'm looking into the audience, and I have a lot of co-workers here. They still have to come in on weekends to look after their cells. But I think, this is really where engineering comes in. Mm-hmm. Yeah.
Colleagues, reaction on the throughput?
Enormous advances in the recent years. Now we used to have plates with maybe, like the throughput used to be 8, let's say, wells where we could do a complex tissue on a plate, and now we have 384. We really went from small 384 well plate scale. We have also bioreactors to grow organoids more and more. I think the scalability is there. Standardization, perhaps not yet. I think you mentioned standardization in your question. I still see lots of. We have our own system, our own QC, our own methods, and we don't have like a clear SOPs in the community at large. And to refer back to the animal models, I think one limitation of in vitro system is we go one tissue or one organ at a time. We can scale that one tissue. However, it's one tissue, it's one organ. In the mouse, you can collect 20 tissues of interest in one mouse. So these are the differences. But overall, I think it's worth continuing the investment in scaling and standardization.
Mm-hmm. Yeah, I think a lot of the technology comes out of academic research. And when we do academic research, and I remember this from my time as a PhD student in Neuchâtel, you want to try all the different technologies, right? You want to optimize your microfluidic system to be just the best out there. And you don't really consider much about automation, scale up, compatibility with industry standards. And so a lot of the inventors of organ-on-chip systems, I think, all go through that same cycle, right? They have this wonderful system that they generated during their academic research time, and then you meet the folks at Roche and Novartis, and they look at this and like, what should we do with it? This is all super experimental. And so making that transition at this point to say, well, the core technology that's in this chip is really fantastic, but now we have to adapt it in a way so that we can scale it up, that we can go to 96, 384, well, maybe 1536 at some point. And we are making this compatible with the workflows and the instrumentation that is already available in the pharmaceutical industry. I think this is key for success, because that's when, I mentioned democratization at the beginning. If every experiment starts with a 1 million investment into a machine that nobody has, then that's a big hurdle for adoption. But if you can bring this into the workflows that are already established in the industry, that can really help. And then that's also where quality comes in. So for me, automation is not just throughput, it is also just better quality of the data that you generate, because you take out the human factor to some extent. And it's simple things, right? Pipetting the same substance on one plate in 1 second or over 5 minutes, because you take the time to go through the rows and so on. That can have an impact on the data quality. And so the more we can run this automatically, the better the data that we're generating is going to be.
Yeah, and of course, with all these new technologies, sequencing, imaging, we generate a huge amount of data. Now we speak about the parallelization. We need to put also the digital twin in the equation for both MPS, but maybe also for early stage of human trials. Mm-hmm. What do you think?
So I think the fact that you can actually get a lot of information out of an organ-on-chip system is fantastic, and it's scary at the same time, right? So the one question is, what do you do with all of this data, and how is it actually guiding the decisions that the researcher needs to make every day, prioritizing compounds and then pushing them through the pipeline? I think that is a big concern, but this is where machine learning can really help us. If you consider an organoid, it is a mix of very different cell types that are all relevant to the target tissue. But even these different cell types are not, or the groups within these cell types are not the same. A cell that lives on the outside of a sphere has a different life than the one that lives on the inside. It gets more oxygen, it gets more nutrients, but it's also affected by the drug of interest at an earlier stage and maybe at a higher dose. So how can we harness this non-uniformity, this diversity of information that we have in these models, and how can we then fuse that into actionable insights that allow us to take decisions on which drug we believe is going to be more efficacious, or put less of a burden on the human body? That requires a lot of data science, and it could down the line lead to that human twin. At the moment, I would rather see it as a tissue twin, so to speak, right? Because that's really the level of depth that we have taken at this point. But the wet biology needs to work together with the digital analysis of this information. That is a strong combination. If you just try to do one thing, then you're going to fail down the line.
Any reaction on the digital twin?
Not much. I think, data generation analysis has to go hand in hand. We generate a lot more data per experiment than previously. And so with that, you need to up the game on the analysis side. And really that's a cycle, because it allows you to design the better experiment next time you run it. Mm-hmm. So I don't think there's a lot to add. Mm-hmm.
Mm-hmm. Regulation, we already mentioned it, that of course it's critical to ensure the accuracy, the safety, the reproducibility and ethical compliance. And when we have to shift away from the animal models, of course, some entities have to take the lead to have to bring some pressure on the regulatory authorities. Should it be from the end user, from the biopharma, who depends from the validated tool, or more from the techno provider developing the platform. And we have the chance to have both sides. So happy to have an animated debate here.
It's an end-to-end, I think, is the solution. We clearly need, so what the FDA Modernization Act 2.0 has done is to provide now a regulatory framework. So that now we have. It has not changed much on how we probably were approaching this anyways with the guidance, but to have now the legislation that supports what we are proposing can be a game changer. We have to get there. And I think this is then the one end. And to work together with the regulators, understand what they are willing to accept, to evaluate in an IND submission, and to see if there is a preferred technology they would evaluate. They have what they call the iSTAND program, evaluating technologies and making sure that these technologies are validated. The authorities call the NAMs, the new approach methodologies. And I think the technology developer starts, and we are, I think in pharma, the end users internally. So we directly interact with the authorities. So I think this end-to-end interaction, and we've seen that working well. I think it can work better. And that relates to then the standardization, to the robustness, to building the confidence in these models all together. With the specific use of case of use, as you mentioned.
Yeah. Thank you.
I would very much agree. From a technology provider position, the regulators play a very important role, but we can also overemphasize the role. I think I probably speak for every startup founder in the field. I think none of us has not at some point in the past said to their investors or board of directors, if FDA would endorse our technology, we would already be making millions and millions. Of course, that would be fantastic, but ultimately it's not the FDA's role to endorse a specific product, and neither for EMA and NIEHS in Japan and so on. So the regulator is the referee, right? And we are the soccer players, and we have trainers and we have players. It's our job to win the game. It's not the job of the referee to win the game for us. Can the referee make things a little bit easier? That's absolutely the case. One of the things that I think would be very helpful for us, if there would be more guidance that helps an end user to understand whether a specific technology is fit for purpose for a specific application, for a specific context of use. How good does it need to be? What kind of biological features does it need to show? How does it need to respond to a certain set of test compounds? I think that would be possible, and we are exactly going in this direction. So iSTAND, that Annie mentioned, I think is a great approach, and we see that regulators are taking more interest in this technology. So just a couple of days ago, the FDA published a paper using our liver microtissues to test antioxidants that are part of rubber tires, to understand the health risk of this contamination. And they used it as a product, and they liked it, and they presented the data with it. That's the kind of endorsement that is very helpful, but that's sort of our job as technology providers, to have solutions that help people to do a better job. It's not the FDA that's going to do that heavy lifting for us.
Yeah. Dominique, would you like to add something?
It's just an ongoing conversation. I think, the FDA 2.0 Modernization Act just signals the willingness to look at certain things in a different context. It's like you mentioned with the soccer game, once you no longer play with a ball, but with a different thing, then a lot of the rules don't make sense anymore. And what I took from the Modernization Act simply is that the authorities accept that, okay, if you come with novel entities that have different characteristics, some of the tests that we have been asking in the past may actually no sense. And if you can convince us that you actually have built something that captures certain things that we want to make absolutely sure you tick the boxes before we allow you to go into patients, okay, we're willing to discuss, convince us. And I think, that's the great signal, because that's a scientific exchange. And it's not just, okay, you need to do this, and then we don't care if it makes sense or not. I think that's, for me, the positive aspect about this.
Yeah. Thank you. So before taking some question from the audience, maybe a few words for the future. Now we discuss about the automation, the AI. Of course, we would like to integrate everything. But what's your sensitivity or your feeling to unlock the full potential of the MPS in the next, 10 or 20 years? How would you rate these different topics?
Kind of what if I had 3 wishes with the good fairy? Okay. Well, immune component, vasculature, and aging. Because a lot of those organoid systems that we create from induced pluripotent stem cells, once they have reached their target tissue, they're actually really recreating embryonic tissue. And again, I'll take it back to neurodegenerative diseases. If you have a cell that you now create, even if you can create something absolutely wonderful and fantastic like a cerebral organoid, that's like the cerebral organoid of not even a yet born baby. And so now asking, hey, what about Alzheimer's? It's pretty pointless. So that's definitely something that we have to work on. Plus the already discussed immune system, and then again vasculature to also touched on things about having not having gradients in terms of nutrients and oxygen, and again having access of the tissue to other things.
You mentioned democratization, and I think this is also less technical, maybe more philosophical now, but democratization in the sense that to make those bioengineered system MPSs accessible to all, small companies, large companies. We are now in an era where we still have to run both the conventional animal testing and the in vitro system, because there are still uncertainties when we file with when we submit an IND filing, and this is high cost, high timelines, and not everyone can afford this. I think big pharma can afford this. Smaller companies, startups cannot really go this way, so they really, of course, opt for the lean package for submission. So to me, this is still now a luxury that we can do all these activities in companies like Roche and Novartis. And I think this is a limitation for adoption. It's a limitation because we need more use cases to learn, to improve. We need more end users. We need everyone. So I think my wish, if I had one, would be that there is a very generous sponsor out there that put together enormous resources, and everyone can go and test their good ideas in these systems, and we all learn from it.
Interestingly, that you mentioned adoption. So sometimes it's actually the smaller biotechs that adopt NAMs or MPS faster than the big companies, just because there's a little bit of less legacy and a different mindset. So cost absolutely plays a role, but then also sort of the feeling, hey, let's change things, let's break things a little bit, let's take the risk. That is something that we sometimes see more pronounced in smaller companies. I'm born in the 70s, early 70s, and that was the time when seat belts for cars came up, and I think everybody sort of hated it, right? So some people hated it because they thought they would not be able to get out of a car when they drive it into a river. And then the other hated it because they couldn't afford it, and it was not in their cars, but in the cars of their department manager. And but now we are used to it, right? It's there, and if I get into a car where the seatbelt doesn't work, it really freaks me out. And so that's how normal it became. And so I think if we think 10, 20 years into the future, maybe more 20 than 10, but I think that we'll have a same feeling about some of the the workflows that we have and that we rely on today. And our kids are going to say, did you really believe that that's going to work, right? But it's just the legacy that we have. So I think we always believe that we are at the pinnacle of innovation, and from now on, things are pretty much going to be static or slowly incremental. But I think that's what every generation in every domain thought for the last 200 years. So we will see a lot more progress in this field, and the combination of data science and advanced in vitro models, but also advanced instrumentation, advanced medicinal chemistry to develop molecules and produce molecules that are still unusual for us. All this combination is going to give us huge opportunities in the future.
Mm-hmm. So if now we go a bit for specific example, as we have also expert in the audience on specific disease. Do you think that some disease are more ready to benefit from MPS, or some other where it will be difficult to replace some animals? I mean, in the short term. Maybe we do also a round on this topic.
Mm-hmm. That's a difficult one. Mm-hmm. Multifactorial things will, by definition, be much harder to model, and then we're back to the immune system. Yeah. Monogenetic, dominant gain of function, direct cell autonomous things, cancer, degenerative things, I think that's the first front line that we can tackle, and there has been immense progress. I think with stem cell derived MPSs, we can start to really look into cell fate decision, regeneration, abnormalities in development, etc. Again, the multifactorial things, when more than one insult has to come together from different organs, that will be hard. And there we still have to work hard to make that happen.
Yeah.
Yeah, agree, which means most of the chronic diseases we have. Polygenic, polyfactorial, endocrinology, that's difficult to reproduce in addition to immune system, aging.
Yeah. And unfortunately, again, this is where a lot of solutions are direly needed, because we have a rapidly aging population that faces a lot of these things that you just mentioned.
Yeah.
Yeah, you mentioned the aging part, right? So when we create disease models, we typically try to induce a disease phenotype in a couple of days. That typically takes 10 years of insult, right? We are quite strong in the metabolic disease space, type 2 diabetes, for instance. We can turn islets into a diabetic islet within 2 days, 3 days. And then we try to cure it with the drug that we test in another couple of days, right? So the big question is, what sort of diseases can we faithfully capture with such an accelerated approach, and where is it going to fail? And so understanding where a model technology can actually do the trick, but also being acutely aware of its limitation, I think that's very important, especially if we look at the commercial aspects of MPS, right? So we are trying to sell our solutions, but we need to be brutally honest about what it can do and what it cannot do, because otherwise we are killing this emerging market by creating overinflated expectations and then under-delivering on those expectations. I think it was a physicist who said at some point, all models are wrong, but some are useful. And I think this is sort of what we have to keep in mind, right? So we are deliberately simplifying a very complex human body. And if we are just focused on what the model can do, but we are not as much focusing on what it cannot do, then we're doing something wrong.
And it's probably a true opportunity there, because this is also what we do with animal models to accelerate disease. We really have accelerated pathogenic mechanisms that just don't exist in humans anyways. And we know from vascularization of organoids that this is how you can also make them more mature. You can also increase longevity in vitro as well. So there are advances really pointing in the right direction that maybe not aging per se, but at least longevity of a certain organoid. And you start with maturation, and then you can keep them happy for a while and then unhappy for the other while in a dish.
Yeah. So you mentioned, Jana, with the seat belt, so it's now belong to an old time, but also in the in vitro models, the attention that we put on the diversity, especially for the gender diversity in the MPS. Could you comment how do you integrate this part in your system?
So we did a lot of work on diversity, and we still do a lot of work on diversity. Diversity was a great word a year ago, and then suddenly, especially in the US, diversity became kind of a not word.
Yeah.
And we decided, are we going to change this to vulnerable groups, or are we going to stay with diversity? And so we decided to put out a blog on how important diversity is for science, because we are not the same. As you said, the T cells are not the same in the individuals, which are not the same. If we start ignoring this, I think, again, we're making a big mistake. And then at the same time, the more precise an intervention is, the more relevant it is to understand which patients are going to benefit from it and which patients will be non-responders. And that's only possible if you try different individuals. And again, it's a huge benefit for MPS. If it's scalable enough, you can test a drug on a number of different individuals and understand not only whether it works in an individual, but using single-cell sequencing, even why specific cells respond to the drug and others might not. So it's a huge potential if we harness it. But what we need is scalability, we need cost efficiency, and the big way to get there is cryopreservation. That's an area that we are investing a lot in. How can we make dozens or maybe hundreds of different models for different individuals and make them available at the snap of a finger? And the only way that we figured out this would be possible is by producing it in large quantities, freezing it, so that you can take it and then do your experiment within a couple of days. And so that is a bit of a vision that we have. We are not yet there, but this is a way to get diversity into our daily research routine.
Yeah.
Yeah, diversity in terms of sex and ethnicity. We looked at, and we mentioned IPS, so stem cell derived models could be a solution for scalability, and they are easy to self-renew, but we need to invest in their differentiation. And they could, or we could have here diversity included in the pool of iPSCs that we use as a starter. And groups look into, okay, what's available in biobanks right now? Yeah, stem cell biobanks, to realize that it's, I don't remember the numbers exactly, in the 90% white males. And there is also a silent genome, so many populations are just not represented at all in genetic database. So we have a long way, I think, to go to be also inclusive even in our in vitro system. And there's an absolute need for it. You mentioned also SNPs in different metabolic enzymes. This is also true in sex differences and ethnicities, and we know that. And we know that some drugs are not metabolized in the same way in different subpopulations, yet we run the clinical trials in exactly the same way.
Thank you.
I think, diversity for drug discovery, it's a privilege and a need and a responsibility, and I can quickly talk to all three. So it's a privilege because 50 years back, we simply didn't have the tools to do anything in this direction. You had HeLa cells, and again, you did whatever research you did on mammalian cells, you did it on HeLa cells, because that were pretty much the only cells you could cultivate on plastic. Now, at one point, we felt like, oh, it's fantastic. Apart from HeLa cells, we have 20 other cell lines. Then came the revolution with induced pluripotent stem cells. And again, the privilege comes from all the real-world evidence, all the sequencing. We started to realize, oh my God, there is so much diversity, all these SNPs, and they're not represented in the 2 cell lines that I have in my lab. But first you need to understand in order to now correct it. And I think we see a lot of effort nowadays to correct this in our lab. But again, first you need to have the means to actually correct it. You need to see it. Before we were simply blind. Now we're no longer blind, and now we're responding. But again, sometimes it's also hard to source in these cells. But there are really interesting concepts also with village in addition, things like this. And finally, with that comes the responsibility. Now we know. And now we're responding, because eventually if we want to make drugs in our pure own interest also, we want to make them work in the majority of those patients out there. And if they don't work in some patients, we want to understand why not, so we can actually tailor a drug to these patients. And so, for me, that's a full cycle. But it's not something where just one day somebody finger pointed and said, you're not doing diversity. It took a lot of learning and understanding and then capabilities to actually address it.
Thank you for this input. I already took some questions from the colleagues online. Do we have questions in the audience? Yes. Maybe with a microphone for the people who are online. Thank you.
Thanks for the presentation. Maybe really a spontaneous question, because like you mentioned Tila cells, which like 20 years back, apparently you have been in the other direction, not diverse, right? Only female cells. Now we're having only 90% male white male cells. I'm wondering why. Like, are we discriminating in cell lines, or is it the people who are ready to give their cells? Or why? What's the reason behind this?
Yeah. For sex, I would not know exactly, but definitely for ethnicity, it's about where the research is happening, where we sample, and also there is reluctance also for certain populations to participate to research and donate for obvious. I think for reasons we can definitely understand. So yeah, I think it's where research of that caliber is happening is mainly biased towards Western countries and then white ethnicities.
We have one question in the back.
No, just a comment why.
Just wait.
The reason is easy, because the male iPS cells were more stable. Genomically they were stable, and the female ones were not stable. And that's why everybody was working with male iPS cells.
That's true.
But now everything is solved, so we can also work with females, and it's coming. Huh?
Thank you.
Thank you.
Here's a question. Yes, just here.
Hi, thank you very much. I'm here for the interesting discussion. Just a follow-up on the sex differences again, because males from females have very big systemic differences. Like females have all these hormones, all these different regulations. So will that be ever addressed in in vitro preclinical model, you think?
Mm-hmm. So we are actually working on a diversity project where we are changing the hormonal composition of the media, cell culture media over time to mimic this. It's too early to say whether we really have translational data, but that is one aspect. So, the cell source, but then also creating the environment for the cell or the 3D model to grow in the right way. Mm-hmm. That is one of the ways how we try to address it, but it's still early days.
And maybe to add to that, that's another great example where actually the whole in vitro and MPS technology is actually advanced understanding. So women's health for a long time was very much understudied, or is very much understudied. Sometimes, but also because it's very hard to get consent, or you definitely don't want to interfere in pregnancy, etc., with doing some science. Thanks to iPS-derived endometrial and placental organoids, this is a subject of study by Margarita Turco here at the FMI, where you are, yes. So with that, that's, again, it's once the technology advanced, we actually enables this type of research. And I really would hope that, again, also a lot of these blind spots now get attention because suddenly we have the tools to actually start to look and shine some light on.
Yes.
Thank you so much. Thank you so much for the talk. When I was in university during my studies, I actually worked on microfluidic systems. And so I saw a lot of the development that the universities are doing in regards to this. However, I also heard a lot about how difficult it was to adapt it into industry. Now, you shortly touched upon this, but I was curious, do you think it's good that universities are trying so many new ways to get microfluidic systems and small cell cultures to work, and that it doesn't really get adopted into industry? Or do you think they should collaborate more and universities should focus more on getting it really to work on big scales and not just for themselves?
I think that's a fantastic question. So I did my PhD in the early 2000s on a microfluidic system, and the story that was out there was microfluidics is the same as microelectronics, right? So there will be Moore's law, complexity or density of channels is going to double every year and cost is going to halve every year, and this is an exponential development. I think at the end of the day we can fairly say that this hasn't happened, right? So there are very few microfluidic products out on the market that really made it into a mass market, like inkjet printheads and stuff like that. So I think my advice would be for academic research to talk to the end users of if you are in a translational project, right? If it's basic science, then it's a different story. But if it's translational science, to talk to the end users that are supposed to use this technology early on to understand what the needs are, what the boundary conditions are, because then the academic science can be more impactful at the end of the day. Maybe it gives you blinds as well, and you just have tunnel vision on developing something which is not necessarily the job of an academic researcher. But early on understanding what is truly needed. For me, it would have been fantastic to see my PhD research to become a technology that people are using, which never happened. I had great papers, great conferences, and so on, so I'm not regretting anything. But the true satisfaction of creating something that helps thousands of others to do a better job, I think that is fantastic. But you need to understand how these 1,000 people want to use that technology from early days.
Okay, let's take one question here.
Thank you for the excellent panel. My question is about the vascularization. So it's a holy grail in tissue engineering and any kind of tissues in the lab, basically. And my question was more on as well that the longevity of disease is really difficult to study. But did you also see that there's a limitation to study many different disease models depending on the oxygen need, for example, of the model? For example, islets, they need a lot of oxygen. So I would see also that they're very challenging in the lab and they're very demanding. But would you see that now the current models prefer some tissue types over other ones because the oxygen delivery is really difficult without vascularization?
So we see a little bit more the other way around. Like classically, you think like 21% oxygen, that's the norm, but actually very few tissues in your body experience this partial pressure of oxygen. So we actually, again, sometimes you feel a little bit stupid, but now we've learned, okay, if you dial it down, that is where your system actually starts to mature much better, and you can keep it in culture for much longer, and you get much more relevant answers. So I think, yes, vasculature is one thing, but again, just to ask a very simple question, what is the physiological environment that you want to mimic? And again, these things have so many parameters. I don't want to blame anybody, but sometimes you just overlook obvious things.
And can I just add maybe to this in terms of what can be done? And you may think it's very complicated and not feasible to have a vascularized organoids. What we learned by trying to grow organoids in different conditions, if you take, and that's close to what I do in research, kidney organoids derived from stem cells, from iPSCs. if these organoids at a certain stage in their development are exposed to shear stress, mimicking again the tissue environment, there is a potential of endothelial cells within these organoids, and then we start seeing blood vessels, micro blood vessels emerging from within the organoids inside. And it's not a mature vascular network that you have, but at some point we could get these organoids by shear stress, by matrix and chip. We were able to see movement of immune cells within this newly vascularized organoid. I think this is amazing. I think this is truly true, and we saw that it's truly amazing when you see that happening, and to have immune cells circulating within this organoid also contributed to a biological response from the organoid, and we saw interactions between the endothelial cells and immune system. So it's complicated, it is complex, but when at some point you try and you see that biology finds itself and biology works, so we need to try. Yeah.
It's spontaneous. That's.
Mm-hmm. Yeah. But I think we were lucky because it's not all organoid types that work like the kidney.
Gilles, there's a question.
So we will take the last question for the person in the room. Let's take here. Just wait for the mic, please. Thank you.
Thank you so much. So Jan, you mentioned that when the model is perfected too much, too early, it generates a lot of data that ultimately nobody is gonna use. And if it's too simplistic, then it produces wrong results. So I wanted to ask about the metrics that come into play for knowing where the line is and when to stop perfecting the model.
Yeah. That's a great question. So we look at this a little bit from the topic of quality engineering, right? So in quality engineering, you do exactly this. You want to figure out if you have an airplane, how much preventive maintenance do you have to do? If it's too expensive, then the airline gets bankrupt. If it's too little, then the airplane falls off the sky, right? So both are sort of undesirable outcomes, and you want to be in the middle. So what we look at in terms of cost is not just the cost of the platform, the cells, the microfluidic chips or the plates, the assay reagents, but also the whole cost of ownership, right? How long does it take to culture a system? What's the yield? How often do you need to repeat experiments? This is all things that add to the cost. And then the cost for not being good enough means higher attrition. You can measure this, I think, to some extent, but there's also a risk for patients. There's a risk for test subjects in phase 1 clinical trials. This is a lot harder to put a number on, right? Because it could be devastating at the end of the day. So I don't believe that there is a formula that you can run and then take the perfect decision. But the more important point is to define the scope of the research, the context of use properly, to understand how many compounds you want to test, how much money you're willing to pay from an end user perspective, and then pick the most simple but yet complex enough model to do the trick. And we see that end users are getting more and more knowledgeable, more and more mature to take these decisions. 5 years ago, you just wanted to try technology, and now you're looking for technology to implement for good, and I think that's a major change in the market.
So thank you for all the interesting questions. I wanted also to touch the innervation on chip with the brain organoid intelligence on chip to finish with a philosophic question, but I think it's already the end. So we need to innovate in the Basel area. We have this unique ecosystem with the biopharma, the clinics, the startup, the academia, and we need really to capitalize and to push this collaboration at scale. I would like really to thank you all for your attention, and especially our 3 great speakers, and we will remain all available during the apéro, and I give now the word to Ursula.
So thank you all. been a very, very enlightening conversation, and I'm gonna take on a point from John, actually, on how the work from academia to startups, they need to look at adoption and look at workflows, because that's exactly what we try to do at day one with the accelerator. We support and enable tech startups that enable pharma R&D, and we try to help them on their path to actually do that on our non-for-profit basis. So I'm gonna hand over to Rahel, and we're gonna close very quickly.
Yes, absolutely. A few words. We operate an office space on Novartis campus for health tech ventures. If you're interested, please come talk to me. More information on the QR code. And yeah, you find me at the apero.
And for everyone on site and online, I mentioned it briefly at the beginning, so open mics will be continued in 2026 on a quarterly format. So we really thank Novartis for supporting us on making these events happen and bringing relevant topics to the pharma R&D industry to a wider audience. So if you are not yet registered in the newsletter, please do. We cannot send you email invitations unless you actively register. So I get a lot of emails on, oh, but I didn't see the event. Please register, else I cannot reach out to you. And on a better note, we have in exactly 1 month our innovation showcase. So that's kind of our public big demo day at the other side of the Rhine this year at the Roche Towers. And it's 2 full days of health tech innovation. A first day, a lot more focused on pharma R&D. We will have panel discussions, keynotes from players like NVIDIA, but of course from Roche partnering, Roche R&D, Accenture, and a lot of startups coming from our cohort on a startup competition. All ticket proceeds go fully to the Startup Award, so if you join, you also contribute to the better good. And just for you to know, if you fill out the satisfaction survey today, you will find the QR code around, and on your email later on, we will give out 5 free tickets to the people or among the people that fill out the satisfaction survey. So your feedback is important here, a bit of encouragement to get the survey filled out. And I think with that, it's time to close it up. You'll have the speakers during the apero might approach for questions and to continue the conversation, and really looking forward to see you very soon again. Thank you.
Thank you.